diverticulum or outpouching from the caudal wall of the yolk sac that extend into the connecting stalk. I'll show you a figure and we'll come here again. So this is the, this one is the embryonic disc, yolk sac cavity, and the yolk sac cavity and amniotic cavity. And this yolk sac, this yellow color is the endoderm, red one is mesoderm, and then the ectoderm. And this one is the allantois. So it is a sausage-shaped diverticulum or outpouching from the caudal wall of the embryo, and it extends up to the connecting stalk. I told you that the connecting stalk is filled up with extra embryonic mesoderm. It is filled up with extra embryonic mesoderm. So I told that the allantois, it appears in 16th day. It is a sausage-shaped diverticulum from the caudal wall of the yolk sac that extend into the connecting stalk. So we see this one, and then the Dorkanai next. So in humans, the allantois remains mostly rudimentary. There is no even full function of the allantois, and usually it disappears. But if it remains, there are some clinical conditions. I will talk it today, few sentences about it. So it is involved with early blood vessel formation in human embryo. That is not very important. Just few blood vessels form around this. So it is involved with early blood vessels formation in the human embryo. So, and look here. So some blood vessels form here. And I told you in embryonic life, where there is mesoderm, blood and blood vessels can form de novo. So some blood and blood vessels form here. So in human, the function of allantois is not that much, but it is involved with early blood vessels formation here at this region near to the connecting stalk. And ultimately, these blood vessels will become the umbilical blood vessels. So the blood vessels of the allantois become the umbilical arteries and veins. We will see later on. They will form the umbilical arteries and umbilical veins. So here will be our umbilical arteries and umbilical veins here. And this is connecting stalk. And the connecting stalk connects the whole thing with the mother's uterus wall. So this connecting stalk will become the umbilical cord. Later on, we will see this connecting stalk, which is filled up with extraembryonic mesoderm. And allantois develops near to the developing urinary bladder. So this allantois, it develops near to the developing urinary bladder, because later on we will see that our gut tube will be formed by this incorporation of the wall of the yolk sac. When the embryo will fold, this yolk sac lined by endoderm will be incorporated into the embryo, and it will form the gut tube here, foregut, midgut, and hindgut. And here the urinary bladder will develop, like here. So this allantois develops near to the developing urinary bladder. And as the urinary bladder enlarges, the allantois becomes the urachus. The name of the allantois changes and becomes the urachus, and which is represented in adults as the median umbilical ligament. So this allantois will become the urachus, and later on it will form the median umbilical ligament. I think you know about this median umbilical ligament. And so let us go, and so here will become the median. So first of all, there is the formation of allantois near to the caudal region of the embryo, near to the development of urinary bladder of human embryo. And when the urinary bladder enlarges, the allantois becomes the urachus. And when the baby is born, the urachus form, it becomes a cord. No lumen is there, and it forms the median umbilical ligament. So you know about this median. So in the exam, when you say about the median umbilical ligament, I will ask you from where it has developed. So your answer should be, madam, uricus, and before that it was allantois. Go back. Now, some clinical correlate about this. When the lumen of the allantois disappear, but if it remains or some part of it remains, there will be formation of cysts. Cyst means an empty cavity filled with air or fluid. Cysts means an empty cavity filled with water or fluid or air. So allantoic cysts. Allantoic cysts are the remnants of the portion of allantois and are usually found between the fetal umbilical vessels, and they can be detected by ultrasonography in the uterus. So the allantoic cyst, if the allantois remains, the lumen of the allantois remains, there will be formation of allantoic cysts. The allantoic cysts are found near to the fetal umbilical vessels, and before the birth of the baby, we can detect it by using USG, that is ultrasonography. So we finish it. And they're usually found near to the proximal part of the umbilical cord, near to its attachment into the anterior abdominal wall. Now, the few things here I will just omit. Now we will switch to the sixth event of third week. That is formation of neural tube, which is important. So this is the sixth event, how the neural tube forms. The process involved in the formation of neural plate and neural folds and closure of the folds to form neural tube constitute neurulation. So it is the definition, or in other words, you can say the process by which neural plate, neural folds, and the neural forms and the neural folds closes to form the neural tube is known as neurulation. So first of all, there is a formation of plate, neural plate, then it folds, then the folds closes to form a tube, which is known as the neural tube, and we call this neurulation. A question, this comes in the exam. Define neurulation, or what is neurulation? Define neurulation, or what is neurulation? Marks will be 2 here. Now, these process of formation of the neural tube. So these processes are completed by the end of 4th week. It starts in the 3rd week, but completes by the end of 4th week. That is, whole of the 4th week is required for the formation of neural tube. But we have seen that our heart, human heart, develops, it starts to develop first and ends during the end of 3rd week. So among all the organs of humans, the heart, that is the cardiovascular system, start to function earlier. And now during neurulation, the embryo may be referred to as a neurula. I told you that during gastrulation, when the 3 germ layers are forming, the embryo is known as a gastrula. And when neurulation is occurring, the embryo can be referred to as neurula. So now, what is neural plate? These are all important and interesting also. So as the notochord develops, the embryonic, you have seen in my last class, I told you how the notochord develops from the notochordal process in the mesenchyme, in the intraembryonic mesoderm. So as the notochord develops, the embryonic ectoderm over it thickens to form an elongated sleeper-like plate of epithelial cells, that is ectoderm, and these elongated, thickened, sleeper-like epithelial cells, this forms a plate, and these plate is known as neural plate. So as the notochord develops, the embryonic ectoderm over it thickens. Let me show you a figure. So here, underneath the notochord is forming. I cannot show you here in bhitore. And over there is the endoderm and yolk sac cavity. And here it lies our ectoderm. So when the notochord is developing underneath, bhitore, the overlying ectoderm thickens, becomes slipper-like, and we call it neural plate. Neural plate. Our primitive stick and primitive node is here. So this is the real embryo in 19 days, 19 dine. So we can see that underneath is the notochord, and here the ectoderm has been thickened to form a plate. We call it neural plate. That is what I said, that as the notochord develops, the embryonic ectoderm over the notochord thickens to form elongated slipper-like plate of epithelial cells, which is known as the neural plate. And now, very important, interesting, the edges of the neural plate. Then what will happen? There forms a groove here. একটা groove হয় এখানে, একটা খাঁজ. And here the edges of the neural plate thickens to form neural folds. And the fold will close here, এটা একটার সাথে লেগে যাবে, and form the neural tube. So this thus, the neural plate formation is induced by notochord. Now, in my last class, I said that as the primitive strict develops, the notochord develops. So primitive strict influences the formation of notochord. So we call the primitive strict a primary organizer, which induces the formation of notochord. And when the notochord is formed, it induces the overlying ectoderm to form the neural tube, neural plate, neural fold, and then neural tube. So the notochord acts as a secondary organizer to form the neural tube or neural plate, whatever you say. The neural plate, we call it neuroectoderm. আমরা এটাকে neuroectoderm বলি, gives rise to the CNS, the brain, and the spinal cord. CNS means the central nervous system. In your brain card, you will see, we will ask you, what do you mean by central nervous system? The answer is very easy. The answer should be, madam, brain and spinal cord. Brain and spinal cord constitutes the central nervous system, and it comes from the neural plate, from the ectoderm. So as the notochord elongates, the neural plate broadens and eventually extends cranially as far as the buccopharyngeal membrane. We don't need it. And now neural plate, neural groove, and neural folds. So we know about the neural plate, and we will know about the neural groove and then neural folds. So on about 18th day, the neural plate invaginates. Invagination means goes inward, I told you. ভিতরের দিকে ঢুকে যায়. So neural plate invaginates along its central axis to form a longitudinal median neural groove, which has neural folds on each side. Let us see the figure here. আমি আবার আসবো এখানে. So when neural plate enlarges, there appears a groove here. The neuroectodermal cells goes inward, ভিতরে থেকে ঢুকে যায়, invaginates to form a neural groove, central axis বরাবর, and the edges of the neural plate thickens to form neural folds. তাহলে in language কি বলেছি, about 18 day, the neural plate invaginates along its central axis to form a longitudinal median groove, which has the neural folds on each side. The character ছবি. Another one. So this is the cross-sectional value. It একটু দেখি আমরা. So here is our ectoderm, মাঝখানে mesoderm. Here lies the notochord, central axis. Here is the yolk sac, and here is endoderm, embryonic endoderm. So what happens? এই জায়গাটা, due to the notochord thickens to form the neural plate. A neural plate, due to invagination of these epithelial cells, it forms a groove, the neural groove. the edges of the neural plate thickens to form এই জায়গাটা আর এই জায়গাটা to form neural folds. Next, the neural folds become particularly prominent at the cranial end of the embryo and at the first signs of brain development. The neural folds become particularly neural folds. These are all neural folds. These are all neural folds. So these neural folds become particularly prominent at the cranial end of the embryo, and these are the signs of brain formation. এখানে brainটা হবে forebrain, midbrain, and hindbrain. এখানে spinal cord. So the neural folds become particularly prominent at the cranial end of the embryo and at the first signs of brain development. By the end of third week, the neural folds have begun to move together and close, converting the neural plate into a neural tube, the primordium of CNS. So if you say the neural plate is the primordium of CNS, is okay. If you say neural tube is the primordium of CNS, is okay, because neural tube comes from neural plate. Neural plate একটা থাকে, then there is a formation of groove, and neural fold will fuse with each other, form the neural tube. একই কথা. So let us see here, look here, এখানে দেখি neural groove and neural folds here. Next, the neural folds are, groove is deepening and the folds are coming close together, and ultimately the neural folds will fuse with one another, and here will be a neural tube like the notochord. এটার মতোই, এটা একটু different, and it is also different, একটু different. So first of all, there is neural groove and neural fold. Neural fold comes close together, and ultimately they will fuse and they will detach from this ectoderm. এটা detach হয়ে আলাদা, so there is the formation of neural tube. That is what আমি এটাই বলেছি যে by the end of third week, the neural folds have begun to move together and fuse, converting the neural plate into a neural tube, and the neural tube is the primordium of CNS. So I'm showing you the figure. এটা দেখাচ্ছি. So ultimately they will form the serially. I can show you neural groove, neural fold thickens, groove is deepening, and there is the formation of neural tube. The neural tube soon separates from the surface ectoderm. That is what I say. The surface ectoderm take a separate. It will separate from the surface ectoderm here. Can take a separation, and the free edges of the surface ectoderm fuse so that this layer becomes continuous over the neural tube and back of the embryo. What does it mean? After the fusion of the neural folds, it will detach from the surface ectoderm, and the surface ectoderm again fuse with one another, so it will be a continuous layer at the back of the embryo. And think about your back also. Think about your back also. So this is how the neural tube is formed. So subsequently, the surface ectoderm differentiates into the epidermis. So the surface ectoderm will give rise to epidermis, because our skin has two layers, epidermis and dermis. And the epidermis comes from the surface ectoderm, epidermis. So neurulation is completed during the fourth week. Start during third week, completes during the end of fourth week. Now, neural crest formation. What is this? Let us see. Now, as the neural folds fuse to form the neural tube, some neuroectodermal cells lying along the crest of each neural fold lose their epithelial affinities and attachments to the neighboring cells. So when, look here again, that neural folds are here and neural folds will fuse. So as they are coming close together, some of the cells here in the crest of the neural tube, crest mane hocche chura, the peak speak of the neural, so some cells, these epithelial cells, that is the ectodermal cells, they are epithelial in nature and they differentiate themselves. They lose their epithelial affinity and they detach from here. To become epithelial cells, the cells are close together, you know, because you have learned it from histology, that one of the criteria of epithelial cells, that the epithelial cells lie close to each other. Yeah. So what is neural crest? This is now known as neural crest. I mean, amar jatchi. The neural folds fuse to form neural tubes. Some neuroectodermal cells lying along the crest of each neural fold lose their epithelial affinities and attachment to neighboring cells. So they lose their epithelial affinities. So one of the criteria of epithelial cells is to they are close together and they detach from here. এখান থেকে সরে যায়. And they detach from here and look here, the neural crest cells, and they will migrate here. Look at the arrow. So they lose their epithelial affinity. They try to detach from here. So these are the neural crest cells. And so the neuroectodermal cells then get separated from the neural folds and form a flattened irregular mass of neural crest between the neural tube and overlying surface of the ectoderm. So what do they do? তারা কি করে? আমরা এই যে they forget about this. They form a flattened neural crest cells here. তারা change হয়ে যায়. They become separated. And the neural crest cells soon separate into right and left parts that migrate to the dorsolateral aspect of the neural tube. Here they give rise to the sensory ganglia of the spinal and cranial nerves. Let us see the figure. Again, দেখাই. So what happens here? The neural crest cells, they lose their epithelial affinity and they will detach. And then they will lie here first. এখানে থাকবে between the ectoderm, surface ectoderm, and the neural tube. And then they will be separated into right and left parts. So I'm showing here the right part. এখানে একটা right, and here will be the left part. And they will form the sensory ganglia. They will form all the ganglia. Here they will form the sensory ganglia, sympathetic ganglion. They will form the pre-aortic ganglia. They will form the enteric ganglia. They will form the medulla of the suprarenal gland here. So all the sensory ganglia, including the dorsal root ganglia of the spinal nerve, dorsal root ganglia, they form here the dorsal root ganglia. So we will ask you, whenever you say about the dorsal root ganglia of the spinal nerve, আমরা জিজ্ঞেস করব from where it has come, your answer should be neural crest. Then they will form the sympathetic ganglia. Then they will form the pre-aortic ganglia. Then they will form the enteric ganglia. Then they will form the suprarenal medulla. So all the ganglia are formed by the neural crest cells. Okay, the sensory ganglia of the spinal and cranial nerves, also the cranial nerves. And neural crest cells migrate in various directions and disperse with mesenchyme. What is mesenchyme? Mesenchyme is the embryonic mesoderm. Also, these neural crest cells, they disperse. They lose their epithelial affinities and they can migrate like mesenchyme. ঠিক আছে, mesenchyme মতো they can move around and they then disperse within, মিশে যায় within the mesenchyme in some regions, not everywhere, কোনো কোনো জায়গায়. Neural crest cells give rise to dorsal root ganglia of the spinal nerves and ganglia of all the autonomic nervous system. That is sympathetic ganglia, parasympathetic ganglia, all the ganglia comes from neural crest cells. So we will ask that এই যে ganglia গুলা কোত্থেকে আসে? Dorsal root ganglia, autonomic ganglia, some of the cranial nerve ganglia, they come from neural crest. The ganglia of cranial nerves 5, 7, 9, and 10, they are derived from neural crest. Ganglia of cranial nerve 5, 7, 9, and 10. 5, 7, 9, and 10, remember. Also, they form the neurolemma sheaths of peripheral nerve and the meningeal coverings, that is the pia mater and arachnoid matter. not the dura mater. Just remember, কারণ আমরা এখন এটা পড়াইনি. We haven't started this part of the body. Just go through that the neural crest gives rise to neurolemma sheaths of the peripheral nerves, meningeal. What are the meningeal layers? Pia matter and arachnoid matter. And also they form our pigment of our skin, melanocytes, adrenal or suprarenal medulla, and several skeletal and muscular components of head region. Several skeletal and muscular, so they mix with mesenchyme, তারা মিশে যায় mesenchyme সাথে, then they form several skeletal and muscular components of our head region. So they form the melanocytes, suprarenal medulla, several skeletal and muscular components of our head region. So question, name the derivatives of neural crest. So if you see the Langman, in Langman, there are the derivatives. They have given a table in a tabulated form. They have written the derivatives of neural crest. There are most probably 15 derivatives. You can go through because it comes in MCQ, it comes in SAQ. So name the derivatives of neural crest. Now, the derivatives of ectoderm. So ectoderm in a flow chart, it is divided into two parts. First of all, the neuroectoderm and then the surface ectoderm. And surface ectoderm gives rise to our skin. What is that? The epidermis of skin. And neuroectoderm gives rise to neural tube. Neural tube gives rise to CNS and neural crest. And neural crest, there are several derivatives of neural crest. There are several. So, এভাবে লিখি, ectoderm, প্রথমে neuroectoderm, surface ectoderm gives rise to epidermis of skin. From the neuroectoderm comes neural tube. Neural tube gives rise to CNS and neural crest, and there are several derivatives of neural crest. I have not given it here. এখানে করি নাই. Mention the derivatives of ectoderm. It's a question. Now, clinical correlate. Congenital anomalies resulting from abnormal neurulation. What can happen? We can understand that neural plate folds and then fuse. If there is incomplete fusion of neural folds to form neural tube, there will be anomalies. Jodi neural plate, the folds do not fuse to form the neural tube, সেখানে abnormalities will be there. Like neural tube defects, NTDs, are the most common congenital anomalies. Neural tube defects, NTDs. Incidence 16 out of 10,000. And there is a condition known as anencephaly. A means no, and cephaly means brain. There is no brain. So partial or complete absence of the brain. This is the most severe NTD and is also most common anomaly affecting the CNS. So CNS is jato anomaly hoy, among them, this anencephaly is common. And this baby, this is taken from Langman. This baby has anencephaly. Look here, there is no brain here. And this baby will die, will not survive. Maybe the baby will die dead, or after delivery, the baby will die. They're not competent for life. There was a question one time that anencephaly develops why? Marks was 3. Explain from your knowledge of anatomy or embryology why and how anencephaly develops. Etar answer ta, this is very easy. The answer should be, there is failure of the neural folds to fuse and form the neural tube in the brain region. There is failure of the neural folds to fuse. Jodi neural fold fuse na kore, and jodi neural tube to form na kore oi jaga in the brain region, there will be anencephaly. So the answer ta khub easy. There is failure of the neural folds to fuse and form the neural tube in the brain region, anencephaly will develop. Now the seventh event, and after that I will finish today. So this is also very important, development of somites. This is the seventh event and third week. So there is a term called paraxial mesoderm. So as the neural tube form, the intra-embryonic mesoderm, I talked about this earlier, that epiblast invaginate form mesenchyme and mesoblast, and some mesoblast form intra-embryonic mesoderm or secondary mesoderm or definitive mesoderm. So when the neural tube forms, the intraembryonic mesoderm on each side of the neural tube proliferates to form thick longitudinal column of paraxial mesoderm. Paraxial means, axis means center, par means away from the center. So in the axis, there is the neural tube. Axis e ache neural tube. Tar pashe the mesoderm thickens, form longitudinal column. We call it paraxial mesoderm. Let us see the figure. আমরা একটু দেখি। ওদের জন্য হবে প্যান্ডেমিকের জন্য, তাহলে না ঠিক আছে। So there is the, look here, এটা দেখো, this is ectoderm, red one is the mesoderm, and here is the endoderm. এটা yolk sac, এটা amniotic cavity. So here is notochord. So when neural tube is forming, the mesoderm thicken here, এই যে axis এর কাছে, and they form the paraxial mesoderm. They form the paraxial mesoderm. So as the neural tube form, the intraembryonic mesoderm on each side of them proliferates to form thick longitudinal column of paraxial mesoderm. এখানে আমরা তাই দেখাচ্ছি in the cross-sectional view. As the neural tube is forming, the mesoderm thickens here, এই যে কাছে notochord, and they form paraxial mesoderm. Each column of paraxial mesoderm is continuous with the intermediate mesoderm, which gradually thins into a layer of lateral plate mesoderm. So this paraxial mesoderm thickens to form intermediate mesoderm, becomes thin, and then becomes more thin to form the lateral plate mesoderm. তাহলে mesoderm differentiates itself into paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. এখানেও একই, paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. So each column of paraxial mesoderm is continuous with intermediate mesoderm, which gradually thins to form lateral plate mesoderm. তাহলে so the intraembryonic or secondary or definitive mesoderm is divided into, from medial to lateral, the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. This comes in exam. It ashe, and the marks can be 3 or 2. So we will ask you in the viva also that divide the mesoderm into its different component from medial to lateral. Answer should be, madam, from medial to lateral, paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. So here I have shown you a mesoderm, 17-day, 19-day paraxial mesoderm. Here the intermediate mesoderm, 20-day, and here intermediate and lateral plate mesoderm. Now, development of somite. What is a somite? I have talked about the paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Now we will go to somite. What is this? It's a key. The lateral plate, it's a path, development of somite. I will not go for it. Now, toward the end of third week, the paraxial mesoderm differentiates and begins to divide into paired cuboidal bodies. Longitudinal, it was paraxial mesoderm to kichilo. It was a longitudinal column on each side of the neural tube and notochord. Now the column becomes cuboidal in shape, differentiates to divide into paired দুইদিকে আছে cuboidal bodies. We call it somites, like the brick eat actor per actor, one after another, the brick. So the paraxial mesoderm differentiates and begins to divide into paired দুইদিকেই থাকবে cuboidal bodies, we call it somites. Let us see. These are the now ekhane paraxial mesoderm, when they differentiate into cuboidal bodies, ekta por ekta thakbe, so we call it a somite. These blocks of mesoderm are located on each side of the developing neural tube. So a block of mesoderm, paraxial mesoderm, cuboidal shape hoye jabe, one after another like bricks, itir moto. They lie on each side of the neural tube. So here we can see the somite. Neural tube hocche, neural tube on each side, and notochord is underneath, niche ache. So 1, 2, 3. So paraxial mesoderm differentiates into blocks of cuboidal mass, here one or here one, paired. So they are known as somite. Look here, real picture of embryo, 20 days, 1, 2, 3, 4, 5 somites are here. And now the somites, these are important. These are like mathematics. So the first pair of somites arises in the occipital region of the embryo at approximately 28th day of development, that is end of 3rd week. So the first pair of somite arises in the occipital region of the, think about your occipital region, matha pichone, approximately 20th day of development, that is end of 3rd week. From here, new somites appear in craniocaudal sequence at a rate of approximately 3 pairs per day until the end of 5th week, when there are 42 to 44 pairs of somites. So first pair of somite appear during the 20th day, and thereafter 3 pairs per day. This is like mathematics. প্রথমটা 20 দিনে, এরপরে প্রতিদিন 3 জোড়া, and until the end of 5th week they go on developing, and last of all there are 42 to 44 pairs of somites. And this is the dorsal view of somites forming along the neural tube. Real mouse embryo. Look at the neural tube here. Amra dorsal view to dekchi. This is epidermis. Ekan theke epidermis ta ekটু ডান দিয়ে হচ্ছে, we have just cut through here. And this is the neural tube. Look at the neural tube. এখানে এটা brain region টা না নিচের দিকে, and these are the blocks of paraxial mesoderm. We call it somite. এই যে একটা দুটা তিনটা 1 2 3 4 5. And here the human embryo. এখানে দেখি 22 days. These are the somites. The neural tube is closing here. This is the brain region. Neural folds are enlarged here. এখানে close হবে. This is the lateral view. Look at the somite here. Block of paraxial mesoderm. Number of somites in pairs total, there will be 42 to 44 pairs. There will be 4 occipital somite, 8 cervical somite, 12 thoracic somite, 5 lumbar somite, 5 sacral somite, and 8 to 10 coccygeal somite. So can you tell me with what it correlates? With the number of spinal nerve, number of spinal nerve. So somites are totally, there are 42 to 44 pairs. Among them, 4 pairs occipital, 8 cervical, 12 thoracic, and 5 lumbar, 5 sacral, 10 to 8 coccygeal. We have 1 coccygeal nerve, but the number of somite is more. So what will be the key hobe okhane? The first occipital and the last 5 to 7 coccygeal pairs of somite later disappear. First occipital, there are 4 occipital somite. First occipital will disappear, and there are 8 to 10 coccygeal somite. The last 5 to 7 coccygeal somite disappear. And now the derivatives of somite. The somites will give rise to what? We have seen that somite comes from paraxial mesoderm. So এখন somite থেকে কি হবে? What will be derived? Number 1, the axial skeleton. We will ask you, what do you mean by axial skeleton? আমরা আবার বলছি, the skull, the vertebral column, and the ribs. So what are the components of the axial skeleton? The bones of the skull, not the scalp, skull, the vertebral column. So what does it mean? The vertebrae and the intervertebral disc. Vertebral column mane ki, the vertebrae and the intervertebral disc, and also the ribs. Don't forget about the ribs. The ribs are the component of axial skeleton. So derivatives of somites, axial skeleton, all the skeletal muscles of our body. Remember, this is very easy. All the skeletal muscles come from somite. All the skeletal muscles of our body and the dermis of our skin. The epidermis comes from surface ectoderm, but the dermis comes from mesoderm, that is the somites. Somites come from paraxial mesoderm, so it is mesoderm, right? These three are the component. In exam, sometimes we ask you, or we just in SQ, write down in short the derivatives of somite. If you write this, okay. And later on, we will discuss more. And so somite থেকে কি আসবে? The somite will give rise to, this is neural tube, the axial skeleton. এখানে suppose this is the back of our body, তাহলে এখানে vertebrae and intervertebral disc. তারপর এখানে they will give rise to the skeletal muscles, and here they will give rise to the dermis of our skin. Not needed here. Now, because the somites are so important during 4th and 5th weeks, that is 20 to 30 days, they are used for determining the age of the embryo. Because I just told you that the first pair of somite appear during 20th day, and thereafter 3 pairs per day until the end of 5th week. So as this is very mathematical, we can calculate the age of the embryo by seeing the number of somite. If there are 1 pair of somite, then we can say the embryo is of 20th day. If it is 4 pairs, embryo is of 21 days, like this. So, and this is known as somatic age. Somatic age of the embryo. So if someone asks you, what do you mean by somatic age? You can tell that by seeing the number of somite, we can calculate the age of the embryo. Why? Because the somites are so definitive in their development, because the first pair appeared during the 28th day, and thereafter 3 days, 3 pairs per day until the end of 5th week, when there is 42 to 44 pairs of somite. So this is taken from Langman, like 20 days, there are number of somite 1 to 4. একটাই হবে, right? So this way এটা দরকার নাই. So we have finished. আমার মনে হয় এটা শেষ হয়ে গেছে আজকে. So I have completed this lecture. In next class, I will again discuss some of the events of 3rd week, and then I will go to 4th to 8th week. That is, I will finish the embryonic period, and then I will go to pharyngeal arches, pouches. So finish this day. Thank you, everyone.
Thank you, ma'am.
Thank you, ma'am.
Bye.